Right, so same reminders as last time. Muddy's Point due Sunday, concept check due Sunday, mini project a week from Sunday. Just because today we're going to wrap up the last topic from Unit 2 and then start introducing Unit 3, just to make that clear that this, Unit 2, which is the stuff that you could choose to explore for the mini project, is, goes up through immunocomputing, which is what we will start with today. So unit 3 is where we start with multi-criteria decision making, multi-objective decision making. So how do you actually, move from one objective to multiple objectives without just adding them all together like you might think you do? And so yeah, that's pretty much it. So any questions about that? Okay. So where we left off was that we had last time we had covered genetic programming for automating software development. So we said that there's linear genetic programming, which you can apply to kind of register-based languages, so basically assembly languages that allow for you to as long as you can write programs in strings, which can be broken up arbitrarily, then you can use a GA to you basically turn that into your chromosome. And so linear genetic programming I presented as actually being kind of earlier than genetic programming. It's actually, I think, there's quite a lot of activity, I think, still on linear genetic programming from kind of a theoretical point of view because of this intron exon business that I was talking about. But then there's genetic programming, which uses tree-based structures like abstract syntax trees that allow you to encode anything you want, because basically any parser for a language behind the scenes is using an abstract syntax tree. So generally speaking, anything that is represented by a context-free grammar, like a programming language like Python, C, C++, Java, etc., you can encode in a tree. And then trees have this nice feature that there's crossover that's easy to define for them, and there's mutation that's easy to define for them. So what we're getting at here is that gradually people have been figuring out how to use these population-based and specifically genetic-based approaches for more and more general applications. So it's not just numerical optimization anymore. Now they're actually writing code, or they're fixing code where you feed them in code and then they make modifications to maintain functionality, but maybe squeeze out additional bugs, find and remove those. And so with that in mind, then we're going to transition to the last part of Unit 2, which is to kind of take that to an even further extreme where you kind of get rid of the optimization entirely and you just embrace the diversity generation to maybe do multiple things at once. And that will help us transition to multi-objective as well. And that's this so-called immunocomputing. And so long story short, TLDR, rather than looking at evolution across long time scales, how do you get different species and all that, the folks in immunocomputing say, what are the genetic mechanisms inside a single body that generate diversity for the purposes of anomaly detection? So the cybersecurity of your body. And so if we can learn from the immune system about how it constantly is looking for threats and detecting them and neutralizing them, maybe we can import those same ideas over in something like a genetic algorithm that will allow us to apply it to, let's say, cybersecurity. So you can deploy the things we talked about today on networks, and you can look for, hey, that's a pattern that we don't normally see on that network. Maybe there's something going on here. Or you can deploy them in machine shops, and you can say, that drill press normally doesn't sound this way. That vibration isn't quite right. Maybe there's something wrong with that drill press. Maybe it's actually getting close to a failure mode, and we need to deploy somebody to go and repair that drill press. And so that's kind of the applications we have in mind. So let's give a little bit of background. So immunocomputing in general is just it's studying how immune systems and networks solve information processing problems. That's what we mean by immunocomputing. And they are focused, the main focus of immunocomputing is on so-called artificial immune systems. So immunocomputing is the big topic. And the kind of darling of immunocomputing is the so-called artificial immune system or artificial immune systems. Which is a little confusing because that's often abbreviated in computer science as AIS. But they're one of the two things that there are two types of there are two big categories of artificial immune systems that we'll talk about here. And one of the two categories is abbreviated AIS in biology. So it's a little confusing that this AIS mimics two things, only one of which are called AIS in biology. But that's just how it goes. So the background here is from immunology. And we're specifically focusing on mostly the vertebrate immune systems. So these are the systems in you and me that are constantly looking for invaders and trying to either coordinate general responses like fevers or targeted responses like antibodies that hopefully end up evacuating that invader or neutralizing it. And so how do they do those things? So there are two, maybe I'll go to a fresh page for this. There are two basic immune systems. So there's two basic immune systems in vertebrates. And the one that is more ancient, that is shared with other animals, is the so-called innate immune system. And so this, so it's also found in plants. It's found in fungus, invertebrates. So basically, what I'm saying here, it is old and conserved. So this is sort of the first animal and beyond immune system was this innate immune system that we still have today. And it is responsible for inflammatory response, um, which to infection. So if you get a cut or something like that, and it immediately starts, if it starts getting really red, that your body starts, directing blood flow to it. There's this general response to this injury. That is what we refer to as an inflammatory response. And so it is a sort of generic uh, um, approach that for lots and lots of things that could cause problems, this will be a good thing to do. So we're gonna put a lot of heat there. We're going to put a scab up and those sort of things. I mean, this includes um, things like scabs and etc. So your skin is actually a major component of the innate immune system. So it keeps stuff out. And so a major then function of this is just the removal of foreign substances. But it doesn't learn anything. The innate immune system it's like this very basic classifier where it detects that there is something that is non-self, and it then starts a generic response to that non-self thing. You might get a fever. So a fever is a generic response. Your whole body gets hotter because most of the things that get into your body that you can't survive at that high heat. So it is just a generic way that if you cook the inside of your body, it puts you at a little bit of discomfort for a short amount of time, but hopefully it flushes out anything that's in there. But it's not like when you're sick with the flu, it turns up to this amount of heat because that's perfect for the flu. When you're sick with a cold, it's this amount of heat. It might be that certain things you have more of a fever than others, but they're not fine tuned to that particular thing. It just might be that you get a stronger response with something over another. But it's the same systems here are the systems that are in place to generally maintain homeostasis. So you get a cut very quickly, platelets come by, and whatever fibroblasts and things like that, and it starts forming the scab that then stops the flow of blood, et cetera, et cetera. And so that is a generic response to this generic attack. So that's the innate immune system. And then we have the other one, which is more derived, the adaptive immune system, which is also, or it's also called the acquired immune system. Which in immunology is also referred to as AIS. And so the acquired immune system, this is something that vertebrates have evolved. Now that people are digging down deeper, there are analogous systems that are being found in other taxa outside of the vertebrates that that achieve the same function, but with different mechanisms. But so this here is a more specific response to a targeted response to certain threats. And so this has immunological memory of specific past threats, uh, and um, and it provides enhanced response to specific future infections. So this is the system that when you get sick with something for the first time, your body learns something about that helps identify that particular thing. It learns a way to neutralize that particular thing, and then it stores a little bit of that particular thing, holds it back so that if you ever if you ever encounter that thing again, then it can ramp up the production of those, and I'll use more specific names here, but of those defenses as needed later. And these two systems can work together. So, for example, when your innate immune system notices something that is not self, that this is weird, shouldn't be here, it actually has the ability to do this thing called presentation. It can present the thing that it has found to mechanisms in your adaptive immune system for them to then trigger this immunological memory so they can start ramping up a specific response. So you can have general responses initially that then trigger specific responses later, and then these kind of take over, so that you then don't have to just count on cooking everything in your system to try to get rid of it, so that you actually can have these targeted antibodies that still have a metabolic load. You'll see that if you are measuring your heart rate or whatever, you're using more energy to produce these defenses, but it's not the generic response that makes you feel sick all the time. It's a specific response that can target a particular entity and take it out, as opposed to just try to take everything out. So that's the acquired immune system. And I keep emphasizing it is highly specific, it is highly adaptable, and and it uses something called somatic hypermutation. And I've already mentioned this last time, this VDJ recombination. So this is basically a process of cell division that goes on during in your normal body tissue. It's not like this is not a process of that goes on during meiosis or anything like that. It goes on. That's what I mean by somatic. So it's not gametic. It's not your gametes. This is your normal body tissue, and it employs something that I'll write down here called VDJ recombination. Where usually when you hear the word recombination, you think about sex. You think about mixing the genes of two parents to create a blended version in the offspring. But your body is constantly mixing from its own version of a random number generator, where it has sections of DNA that just have a whole bunch of diversity in them. And it has this ability via this VDJ recombination, which I'll write down here in a second, to recombine reshuffle all of these highly variable pieces of DNA in a huge number of ways to create a highly variable portfolio of antibodies that it then, the ones that happen to match so-called antigens that are on the thing that's not supposed to be there, then end up getting amplified and then produced in mass so that you can have this response. So this is your body's internal way of using randomness to try to find solutions to problems it has never encountered before. So very closely related to what we see in the genetic algorithm. But here, where your body will use recombination to generate this randomness, we don't need to use recombination to generate the randomness. We'll, in our artificial immune systems, just use randomness. We'll use random number generators, real random number generators, and we actually won't use recombination. We'll see that here in a second. Even though the mechanism behind it all, like if people sometimes wonder about this, how is my body generating like all of these different things? It's not like it's generating lots of copy errors. Like that would be kind of dangerous. Let's sort of like try to have controlled copy errors. Here, there's actually no errors that are being generated. It just is maintained again, like a random number generator is basically a mathematical function that whenever you plug in a value, you get sort of an unpredictable value, which you can then iterate and feed, and then you get a sequence that comes out of that. That's basically what the DNA has maintained, these extremely highly variable regions that can be recombined in lots of different ways and to produce outputs which are unpredictable after the recombination. It's again just a natural random number generator. So that process, again, I called it VDJ recombination. And so that's often written as VDJ recombination. And this doesn't matter so much, but in case you're interested, this is like, I think this is variable. And this is, I think, diversity. And I think this is junction. And this is basically, there are three different mechanisms by way of which you can take these highly variable DNA regions and recombine them to create new antibodies. And your body mixes these three different ways. But again, it's basically just a natural random number generator. Yeah. That's a great question. So if you were to look up these mechanisms here, what you find is that it's you have this bank of random strings, and it can do things like, I'm going to take, I've got a bank of random strings here and a bank of random strings here. I'm going to mix randomly from this random strings and this random strings. I'm going to create multiple copies of this random string. I'm going to take this random string and put it over here, and take this random string, put it over here. So it's just, you imagine any different way you could, it's like you've got this bank of randomness. You take scissors, cut it all up, and then you duplicate a bunch of those things that you cut up, and then you put them into kind of a big urn, and then you start grabbing out of that urn, and that is how you produce these antibodies. It's several strands of DNA that get recombined, but for all intents and purposes, you can think of it as one bank of random DNA that is being selected at random for what is going to be expressed in the antibody to match the antigen. Yeah. So I mean, this is again, this is just basically nature's pseudo random number generator. And so this is why it's sort of called, hypermutation, is you're going to get far more variability out of this VDJ recombination than you would get under sort of just normal mutation. And it's not, and I don't, and I want to emphasize, these are not errors being made. It's actually sort of this process that it generates sort of predictable outputs from unpredictable inputs. And that's what gives you such of the random. That's why it's very much more like a random number generator than an error mechanism. So, like when we think about mutation being made when there's just copies of DNA, in that case, it's like, oh, I meant to grab this nucleotide. I accidentally grabbed a different nucleotide and popped it on there, and that was just a total random error. That was just a mistake in the mechanism here. Here, it's not counting on mistakes. It's actually this kind of process that is everything's being combined sort of with high fidelity, but it's being combined in random ways. So that's why, again, I liken it more to an RNG than mutation. So it's a cool way. It's just a way that nature kind of uses randomness and then filters that through processes, which either, and this is what we're going to talk about today here, either is going to, so some of these things will match. So and I don't want to go too much into the weeds of immunology, but there's something called a histocompatibility histocompatibility index that is that in there's every person has a barcode that varies from person to person to person, and every one of your cells has this barcode on it. And when you generate new antibodies, and I should define antibodies and antigens, which I will in here in a second, but when you build new antibodies, one of the things that has to happen in your thymus, and this is so-called negative selection, which we'll talk about here, is you have to make sure that the things that you build don't match any of your cells, because otherwise you get an autoimmune response. You get an inflammatory response to your own body. And that is not something you want normally. Like, that's something that happens, but when it happens, your body is attacking itself, and that generates all sorts of problems. So before you unleash this arsenal, you have to make sure that it doesn't match yourself. So it filters out that, and that's something that we're going to call negative selection or central tolerance. And there is a set of algorithms based on that. But then once you've filtered out all the self strings, then you've got all of these non-self classifiers floating throughout your body. And occasionally you find one of those classifiers happens to hit something. It's like, oh, I found something. And with 75% accuracy, I'm going to say it is non-self. And so at that point, you need to ramp up your production of these classifiers, because you need to be able to find this all throughout your body and fine tune them so that their accuracy then goes up from 75% to closer to 100%. So that is what goes on in so-called clonal selection. So these two processes, filtering self out, so you don't accidentally, get yourself. So it is a multi-classifier problem that's going on inside your body. You want to make sure that you don't get false positives, and you also want to make sure you have you don't get false negatives. And so the false positive rate kind of happens up here in your thymus. And then the false negative stuff happens throughout the rest of your body, where whenever it gets something that looks like something, then it then starts a new process of generating randomness around the one that it found, so that it can generate antibodies like the antibodies that got the high classification rate, but potentially ones that do even better. And then those ones that do even better will get further amplified until eventually you've got a highly specific classifier for that particular antigen. And those are the two processes that we're going to talk about here. So that's kind of what we're going up against, and inspired by these systems. So yeah, I'll just define that our goal is to build antibodies, which you can view as classifiers, that are formulated to bind to antigens, which I'll say foreign antigens to emphasize that they are what's presented on these things that are non-self. And by bind, this just means, I would say identify. And these here antigens, these are just signals of anomalies and threats. And this is without binding to components of self. And so this, without binding to components of self, this is basically, reduce um, um, I, so here we're trying to reduce our false positives, and then here we're trying to reduce false negatives. And I mentioned histocompatibility. I mentioned every cell of your body has a barcode that is specific to you. That's really important. What would be the problem if we all had a human barcode that we all like? The cell looks like a human, but my barcode looks like your barcode looks like your barcode. What liability would that set up? What would a virus be able to do then to get around our immune system? Look like a human. That's right. So the reason that my histocompatibility markers look differently than yours and yours and yours and yours is so that if we all had one single self string, then it would be easy for an invader to just look like that self string. So what's interesting is all of the different ways in which organisms, animals, vertebrates have tried to make sure that these markers over many, many generations of sexual reproduction stay unique to the individual. And this digs down deep into behavior and things like that. Like they've shown that if you put mice, and actually they've shown this a little bit with humans as well, that attraction between mice is often highly predicted by the sort of cosine similarity or dot product of these histocompatibility indices. So, in other words, if you have a male mouse and a female mouse whose cells look very similar to each other, they often will be unlikely to mate. They're just the male mouse or the female mouse won't find the other one to be very attractive. And the thought here is that when you do recombination, you're effectively generating new barcodes that blend the barcode of mom with the barcode of dad, creating a new barcode. And if the two barcodes are too much alike, then there's a chance that the offspring will end up looking like basically the whole family will end up having the same barcode. And if that keeps going and going and going, then you get a huge group of individuals that can then be compromised by a virus that can look like that barcode itself. So there's a lot of these different mechanisms by which this uniqueness is attempts, random mechanisms, so ways in which you try to randomize this during development, but then also these recombination mechanisms where the behavior of the animal actually is unlikely to mate with others that are too much like themselves. So a lot of cool stuff that goes on and a lot of real interesting consequences. And especially if you think about them from a cybersecurity perspective or whatever, like a password perspective, or you don't want to use the same password on every website. Same sort of idea. Anyways, I'm drifting a little bit here, but that's our main goal. So the terms, these are the kind of the key terms here that we'll see: antibodies and antigens. So the antigens are the patterns that are on the anomalies or the invaders, and the antibodies are the classifiers which are built to find the antigens. So they're like puzzle pieces where these things are one half of the puzzle piece that plug into these things, which another half of the puzzle piece. But I don't want to say it's like a physical thing. Like in your body, it's a little bit is like a physical puzzle piece, like trying to get things to match up. But for us, our antibodies, these could be complex computational processes that have a bunch of parameters, which are random, which we are going to parameterize through a genetic algorithm like process. So it might be a decision tree, a whole set of steps that, like, well, I'm going to interrogate this signal in this particular way through these particular set of steps in order for me to determine if this signal is self or non-self. So, and the parameters of that decision tree, that is my antibody. Whereas the actual signal might just be a time series of data or something like that. So it's not actually like this is one puzzle piece and that's another puzzle piece. These are just two complementary things. That's why I'm trying to say view this as a generic classifier. It could be a neural network with lots of weights that you're end up tweaking the weights to see if you can get this neural network to then classify this signal, which is the input to the neural network, as friend or foe. All right, so questions about that terminology? It's sort of clear. And so we're targeting things like cybersecurity applications or anomaly detection, even in things like manufacturing systems and so on. Okay, so with that in mind, there are two basic mechanisms that I want to talk about here. So one of them are the so-called negative selection algorithms. And so this is, I'll say example one, negative selection. These are also known as clonal selection. I'm sorry, sorry. These are also known, this is mimicking, so I guess I'll put it in, this is mimicking the body's so-called central tolerance. And this is particularly good for threat or anomaly detection. And this is another one of these ones that I mentioned that Stephanie Forrest is a distinguished faculty here at ASU who used genetic programming to create these automated software repair tools like GenProg. Earlier in Stephanie's career, one of the big things that she was well known for were the development of these algorithms. So this is another thing that if you're interested, another faculty member who knows a lot about this stuff is Stephanie Forrest. So, how does negative selection work? I'm going to give a detailed explanation of a clonal selection algorithm because it just looks so much like a genetic algorithm next. Here, I'm going to keep it a little bit high level with negative selection, but please, feel free to ask questions if this isn't clear enough. But the general steps here is we're going to generate random strings. I'll say random strings or random classifiers. And then we're going to eliminate these recognizable self strings or self identifications. So that's where the negative comes into play. And then we're going to flag any future matches um, in the set from the set that remains. Or if I want to draw that out, we've got a random set of inputs, and that gets piped through this filter that filters out these self matches. And this happens in the thymus of the vertebrate body. And then that gives us a bank of non-self classifiers. So the basic idea in your body, basically like in your bone marrow and things like that, it starts generating these antibodies, and then they get shuttled up into your thymus where there's more randomization and things like that. And inside your thymus, it's got a bunch of these self strings that it says if you find any that match, then those antibodies get lysed, they get destroyed. So your thymus has the ability to kill these antibodies. But if they don't match, if they manage to run through this gauntlet, then they get dumped into the rest of your bloodstream, and stuff happens down the line. And the same thing's happening here. We have no idea what the anomalous patterns look like. So we don't know what a broken drill press sounds like. And so, all we can do is train on a good drill press. So we don't know what a network being attacked looks like because we've never seen that attack before. But we do know what normal patterns on the network look like. So you have you have basically a bunch of ground truth on normal activity, and you generate a bunch of either random activity or random classifiers for activity. And you basically see, does that match any of my normal stuff? And if I have a sufficiently rich bank of normal stuff, then hopefully I prevent any of these self matches. Anything that's let go, if it makes it through here, a lot of this stuff will be completely useless. But if you happen to notice like any of these behaviors later on down the line, it's probably not normal. It's probably something that at least is worth a second look. And so that's how this works. And so some of Forrest's earliest papers on this were on things like drill presses. And so they would just have acoustic signals, and they just get a bunch of data on what what the waveforms like of what sounded like from a bunch of working drill presses. And they would just put those into a bank, and then they would have a microphone set up listening to the drill presses constantly. And so that if and then they would have a bunch of random waveforms. Now, maybe you had some bias in how you set up these waveforms. Like maybe there's no human voice data in there. Like maybe there's still something that's kind of drill press like about them. But in general, they're just generally random inputs. If those random inputs sounded like normal, then they don't show up here. But then if later on anything that's left over sounds like one of those sounds like one of the signals you're getting through the microphone right now, then it sets off a red flag and says, hey, can you go and check out this drill press? We've never heard a drill press that sounded like this. And it sounds like some random pattern that we generated arbitrarily. So that's probably not good. And then maybe that means that a bearing needs to be replaced and so on and so forth. So it's not just cybersecurity, there's other applications as well. But that's negative selection. That's the idea that we start out with a high entropy set and we filter out one particular set of matches, and then whatever we're left over with, that is our anomaly detection mechanism. So questions about that? Okay, it's just a general idea, and it is patterned after your body's ability to filter out self matches. Yeah. These could include um, anomalies, normal behaviors, um, and irrelevant. But we don't know what's what. So we just have an entropy generator. We have just something to like, let's just generate waveforms, let's generate acoustics. And then we're just going to filter out the things we know are good. Right. The leftovers would be signs of anomaly. Like if you generated a bunch of random stuff and filtered out things that sounded normal, then it may be that that an anomaly is missing. Like it may be that you just didn't generate this input. This wasn't a broad enough set of inputs. But if something survived this negative selection and then you matched it, then that's weird because you just generated this randomly. And so if you're actually hearing this random text or random string, then that probably indicates something's wrong. Now, it could just mean that you didn't have a big enough bank here. But if you have gathered enough data here to filter enough of these things out, and these are things like that's some of the beauty of these immunocomputing approaches is that whereas in the GA, we talk about it running once and then producing a design. You let it run and how long you let it run might change, but you don't think of it as running continuously. Generally, in these immunocomputing approaches, they're designed that they could theoretically continue to be run for a long amount of time, or endlessly, so it may be that you're just constantly generating new random inputs, you're constantly filtering out self matches, and then whenever you get whenever new things pop through the filter, then you just add them to your bank. You might have a fixed size of this bank, and so you're constantly cycling things out on some circular buffer, so maybe the old stuff gets removed by new stuff, but this could just constantly be running as well. And that's exactly what's going on in your body, is that you're constantly generating randomness, checking for self matches, and then in clonal selection, which we're about to talk about, amplifying the things that match bad stuff, and then hopefully not matching good stuff. Okay. All right, so that's negative selection. The other half of immunocomputing, or big part of immunocomputing, is so-called clonal selection. Where this is, you can think of this as that once you've got a diverse set of anomaly or threat classifiers, how do you fine tune them to specific anomalies, and how do you amplify them when you have to think this is a distributed architecture? And so, maybe that distributed architecture doesn't apply to your system, even though it applies to the body, but at least it's inspired by this idea that you might find a little bit of virus here, but the virus, if you find it here, it's probably everywhere. Well, you now need to get the information all over your body. So how do you amplify that and produce enough of it so that you have enough of supply for the rest of the body? That's again what some of these things are inspired by. So amplification and fine tuning is what this clonal selection is all about. And I hope I'm going to write out kind of pseudocode here, and I hope you can see this is a lot like a GA. I'll say it's like a GA without recombination. And we'll see why we're excluding recombination. So the basic idea is we start out with an input set of antigens, which I'll call S. So at least in our case, we're already given a set of known bad antigens. So these are things, again, it's like we're training a classifier. These are things that we want our classifier to be able to classify as bad. And it's like they might be very, very different. Like it's like if you're training a neural network, you got a cat, a dog, a cow, a mule. All of those you just want to classify as animal, and then all the plants you want to classify as plant. So they're very different looking animals, but you all want to sort of gather them. And that's effectively what's being done here. So these antigens might have a huge amount of diversity, but you want to be able to mobilize a response to each one of them. So your output is a set, I'll call it M, of memory B cells. So there is a type of cell, your antibodies, there's a type of in the kind of antibody team, there's a cell called a B cell, and the memory B cells are involved not only in the amplification, but also they're the ones that hang around for the rest of your life, and then or for a long portion of your life in order to be able to match these antigens. So we're trying to build up this immunological memory. And there's a bunch of things like immunology is such a cool thing. Like there's crazy diseases like I think it's measles that if you get they actually reset your immunological memory. So that's like one of the that if you get measles, like you go through the measles thing, and if you survive it, that's fine. But then afterwards, you'll then start getting every other disease you used to be immune to. And people don't completely understand the mechanism why, but it is very clear that that a disease like that actually has the ability to so confuse your immunological memory to reset it to zero. And that's just, that like, so that is makes it really dangerous, right? So there's just like a lot of really cool information processing type processes here that could be inspired or, that people can look into. So it's a really cool set of systems, these immunological systems. But anyway, so that's our input and output. So what we're going to do first is we're going to generate a random B cells. So again, inspired by the previous slide with negative selection. So you can view these as random classifiers. And then if it helps, I'll just annotate these are like random patterns to match. And so now, again, there's going to be some things that'll be similar to the GA, other things that are a little different, but actually really set us up well to talk about multi-objective. So now we're going to go and iterate through our antigens. So we're going to effectively run a kind of like a recombination less GA for every antigen, and it will gradually build up a set of these memory B cells that are accumulated through the iterations. So we're going to say like, for all antigens, and I'll just call the one that we're focusing here AG in our set S, do the following. We are going to calculate affinity of all B cells to AG. And this you could view as kind of a classifier accuracy. So the term we use is affinity. This is basically how well does this B cell match to this antigen. So you could come up with different ways to do that. If it was simply a pattern matching type of thing, or again, this could be something as simple as cosine similarity or dot product, or if it's a complicated classifier that we're trying to build, then it might be that I run this antigen in, and then it gives me spits a probability out from its softmax or whatever, and it says, I think this is a 73% match. Well, then that would be the affinity. And so you're going to find that each one of these classifiers for this antigen, some of them are going to present, there's a 0%. I don't think it's bad. And then other ones might say 50%. So you basically are generating an equivalent of fitness for each one of these B cells. Okay. So once we've done that, then we select highest affinity B cells. Um, and so what do we mean by highest? That's a hyperparameter. So you pick the top ones, the ones that match sufficiently well. And then we on each one of them, we do proportional cloning. So this is like parents, and this is like proportionate selection. So we are generating offspring that are clones of these B cells. And if your B cell is has twice as much affinity as another B cell, then it'll probably have twice as many clones than the other B cell. So we're just extending what we've already learned about proportionate selection to this slightly different application space. And then we determine we place clones in another set C. And so they just had this giant set of clones. And in this giant set of clones, then what we would do in the GA is mutate them. So that's what we're going to do. So for all clones in C, we will mutate each. And then here's where it gets a little different. At rate inversely proportional to affinity. So if you are a really good functioning clone, you already have near 100% match to this antigen, we're not going to touch you. They're not going to, the mutation, it is very unlikely we're going to make any changes. But if you're a high performing one, but it's still like only a 70%, then we're going to mutate your parameters more likely. So we're more likely to make those changes. So that's something that's a little different than the GA, is that in the GA, this is basically saying if you're a really high fitness, we're not going to mutate you. We can't do that in the GA because we don't know what maximal fitness is. There's always could be another strategy that is way better than this strategy. We don't have a natural 100%, but affinity does have a natural 100%. So, you can 100% of the time you match this antigen. So in that particular case, that's why we can kind of do this inverse proportion here. Is because there's no worry about us like backing off the mutation rate too early because we actually kind of know what the top affinity can be. So that's a key difference as well. And then from here, we determine new affinities. And then we copy all clones into B. Notice we haven't said anything about M yet, but we're almost done. We copy highest affinity clones into M. So that is our output set. And then we replace lowest affinity clones with I think with randomly generated alternatives. And that's it. That is clonal selection. And so this basically is our new population here. but our outputs, our solutions are here. So this is a little like elitism. So I hope you can see how inspired this was from the GA, but these key differences. So it's got this for loop where you're applying this to multiple antigens. So if this is a GA would be like, I want to find the best antibody for one particular antigen. That's my fitness function. And just find me the best classifier for this. But here we've got multiple, potentially very different antigens that we're trying to service all at once. So that's why this is getting a little bit like a multi-objective problem here, is that we want to be able to match all of those. We want to have some antibodies that match one that may be terrible at matching another, et cetera, so that we at least have one that have really high affinity for every single antigen. So, and that's why it kind of goes in here and runs like multiple GAs in parallel with the same population. So as we generate really good clones, then we can pop them into our output set that is just generating these nice solutions, but we maintain a population that we can keep drawing from, and hopefully that population keeps keeps candidates for the other antigens as well. But it might be that the one that we popped into M on this iteration, the next time through, it's going to have such low affinity that it won't be able to make it through here. So the performance on one may not predict the performance on the next antigen. So we're generating a huge population of clones that some of them perform well, some of them don't, and then also a population of solutions. And we potentially could just let this run, even as new antigens come in, or we could just have this run until it settles out, and then we've just got a set of solutions. Yeah. I mean, theoretically, you could, right? I mean, if this B cell was really complex, like this is a deep neural network with lots and lots of layers or whatever, then you could imagine that it would have the ability to encode multiple antigens. And so then in one shot, if you got the weights just right, then you could hit multiple of these. Right. Well, that's what I mean. Like you can definitely train again. I don't need to just pick on neural networks, but you can train a neural network so that it is sort of partitioned, the weight space can kind of be partitioned up so that, like, imagine a convolutional neural net. Well, we haven't talked about those, but if the point is, if you have enough degrees of freedom, you can create a classifier that basically is like a bunch of is a like disjunctive normal form. It's got a bunch of ands. Like if I see red and a chair and a this and a that, then this is a classroom. Or if I see blue and a projector and a this and a this and a that, totally different, that's a classroom. And if you had, enough depth in your statistical model, then you absolutely could get a really high rate to two types of classrooms using the same model and a low rate to other classrooms. But that would need to be a complex model. What this potentially allows you to do is use very simple models, but then use kind of an ensemble of them together. Yeah. Hmm. I suppose you could probably try that. I think that the leftover, the so we got highest affinity. I called these parents. So these are the parents that their offspring are going to the clones. Um, if you have some reason to believe that these ones are a better to draw, but I think the problem is if you do too much of that, then you don't get an opportunity to explore the space. So there's a balance here. So the way in this kind of simple approach here, and there are variations on clonal selection, but in this simple version of clonal selection, the idea here is we're maximizing, we store stuff that's known good, and then we maximize exploration. But if we borrowed, we could say, you could say, oh, well, half of these we'll take from the discarded parents because they were pretty good. They just weren't, that good. And the other half will do totally random. That would give you, that sure would hold on to some of those, and that might be a good thing to do. But if we want to maximize sort of our exploration of the anomaly space or whatever, then maybe we just want totally random ones. Yeah, you want every antigen to have at least one B cell that matches it highly. Yeah. Before select, so I mean, we mutate. So here, these aren't being mutated. They're being mutated here to hopefully create, if you had a B cell that had like 80% affinity, hopefully it'll have a mutated version that will have, and again, because there's inverse proportionality here, hopefully the low ones will generate children which have a better, at least some children that have a better affinity. And the ones that have really high affinity, they won't get mutated very much, so they're almost copied through. Why are you, well, because you determine the affinities after mutation. So we mutate them, determine the affinities, and then in this newly generated ones, we say, oh, well, that's a crappy mutation. And then so these are discarding those crappy mutations. Pretty clear? Okay, excellent. So that then we have enough time for me to then open up then our next unit, which kind of directly relates to this. So this this idea here that this is like so notice a key point here. We don't have crossover. There's a focus on diversity here. So the idea here is we're trying to not create one solution which matches one particular problem. We're trying to create lots and lots of solutions that collectively are a diverse portfolio of possible responses to a diverse portfolio of problems. So the emphasis here is on kind of diversity and multi objective or multi criteria decision making. And so that kind of inspires us into this, our new sort of unit 3, which is we're focusing here specifically on these multi criteria decision making and solution nature inspired solutions. So already we've seen that a GA-like approach, if you basically run a bunch of GAs in parallel, you can basically create a population of diverse solutions where you can maintain that diversity. And we would say, well, can we sort of make that more explicit? So before us that we can get into that, I have to sort of set up some preliminaries for what we even mean by multi-criteria decision making. And yeah, so and what I'll talk about is being Pareto optimality. So it'll seem a little weird, but we're going to transition a little bit in here into game theory. And game theory is a subset of multi-objective optimization, where you can view the um, the players in a game in game theory as actually just different individuals who have different objectives, different agendas. So you can consider, for example, you have a player one has a certain utility. So these are, I'll use the word utility here, which is returned to player one, which depends on a set of variables, say x1, x2, all the way up through xn. And then this all the way goes down to a player in, who has its own utility, which depends on those same set of variables. Now, the way this is framed right now, then we view these as the plays of the N players. And here we view these as the payoffs to the N players. So this is utilities. So the idea here is that every player can, it's like the nth player can control the nth play, but can't control any of the rest of these. The first player control the first play, but not any of the rest of those. But whatever the other players do affects the outcome of this game. And so again, we can start to view this as a multi-objective problem where instead of calling it player one, I can say objective one is I want to match antigen one. Objective N is I want to match antigen N. Well, I've got these plays. Like if I only have one classifier, these might be all the weights of that classifier. And if I adjust certain sets of weights, it's going to make it better for my antigen one objective. But if I adjust other sets of weights, it'll make it better for my antigen N. So we don't have to think of these necessarily as players. We can just think of them as objectives, which sometimes are in conflict and other times aren't. And we don't know a priori which ones are in conflict and which ones aren't. So there are different solution concepts that we can talk about here. And in these social settings, we often talk about the so-called Nash equilibrium. And this is one of many solution concepts to games. And so we are going to transition to the Pareto equilibrium. But I'm going to start with Nash because that's the first one everyone thinks of. All right, so in a Nash equilibrium, then the idea here is that for all I, I'll just write it out, why not? For all I, so for every player, then the set xi such that the utility returned to player I, and I'm going to use a little bit of notation here, which I'll explain in just a second. X minus I is greater than or equal to U of I xi x minus I star. And that here is for all xi. Okay, so what's this crazy notation? What I'm saying here is that to make this easier, this is the ith component of the plays. This is the play for player I. This is everybody else. So rather than me writing out x1, 2, 3, I, 4, 5, 6, end, I just said, well, somewhere in the mix, xi is there, and that's what xi minus I. So this is just this is just our string up here where we're only focusing on I. Star is our notation for equilibria. So so this here means this is at our equilibria. So we'll use that notation. And so what we're saying here is that at the Nash equilibrium, then if you're at the Nash, if everyone is playing the Nash, so the Nash is a social concept. So then what we're saying here is that if everyone else stays at the Nash, but you change, then you will do worse. It's a unilateral change. So this is saying here that effectively, this is saying that there is no unilateral change that will improve the outcome for the player, for that player. So it may be possible for you, player 3, to get together with player 5 and say, if we both do this, then it'll be better for me or better for us both. But that requires you to coordinate with them. And a Nash equilibrium strategy is without coordination. So it's this idea that if there is absolutely no way for you to coordinate or you to get anyone else to do anything else, if they're fixed, the best you can do is just assume that they stay the same and there's no responsiveness there. Can you do any better? And at the Nash, you can't. So you're stuck there. This is not necessarily the best social equilibrium. From an engineering perspective, this is not necessarily the best solution. There might be other solutions that make every single one of them better. But the only way to get there is they all have to kind of agree that they're all going to move there, because the instant one of them moves away, it wrecks it for them all. So so that's the idea here behind a Nash strategy. In networked systems where we can't do community, so a lot of times we talk about greedy algorithms on networks. So you have a bunch of robots and they can't communicate with each other, but wherever they are changes how other robots benefit. Or you have a bunch of computers on a network and whatever they're doing brings some benefit to others in the network, but they can't communicate with each other. Well, often if you design kind of a gradient climber on every individual robot. So, hey, this robot, just change your policy to provide a little reinforcement learning agent and say, change your policy so that you do better. And others will be doing their thing with their policy, but you just focus on yours. And then oftentimes those things will converge to a Nash equilibria. So that's what this models, is when you have no coordination, no communication. It's not just purely modeling like some human problem. It's modeling engineering problems too, because very often we do distribute a bunch of intelligent agents that just don't have the ability to share information, but they can sense their rewards. And so they'll do whatever they can to maximize their rewards. So in RL, there are these credit assignment problems, for example. If this is not something that you've seen before, that's fine. This is not, I'm just sort of adding this in as extra. But, if you have multi-agent reinforcement learning, there's always this question of how do I get rewards into the agents? So if I have a global reward, if I can broadcast it to all the agents, that's maybe okay. But then each individual agent doesn't know if the increase in its global reward is due to what it did or due to what its other agent did. Well, I could just say, all right, fine, I'm going to give everybody local rewards. If you give everybody local rewards, then you do get these conditions where every individual agent evolves a strategy that works really well for itself, but just screws the group. And so it just gets in the way of every other individual agent. So if you've got like a convoy on the highway or something like that, then you've got this one car racing to get in front, because that ends up minimizing its time to get home. But it manages everybody else in the network that has, it manages to slow them down. And that is a Nash equilibrium right there. So these are real problems for engineering, for intelligent systems, as well as human problems as well. So next time we are then going to say that, okay, that's fine, that's the Nash, but are there other ways that we can solve this problem? Yeah. That's a really great question. So I didn't get into that. So this is a static synchronous equilibrium. So it just describing the equilibrium. It's not describing the process to get there. In numerical optimization, like if you talk to Dimitri Bortsikas, for example, or whatever, so his whole career has been focused on how do you do distributed numerical optimization problems where you have some idea of what others are doing, but it's a stale idea. and different ways that you can update that. And so it's true that in the processes that can result in these things, then that all matters. But this is just describing the equilibrium of those processes. So not the actual state of, well, like, in fact, you may not know any of these, but still your reward depends on these. But if you are in a network that communicates, once you're in that network that communicates, how stale the values are that are being communicated from others and at what order definitely matters. But hopefully you design an algorithm that is robust to the ordering. And there's a whole set of classes that we might get into that later in this, talking about some of those things, but there's some great textbooks on that. Berticlus and Taciclus, for example, but in general, in any numerical distributed algorithms course, you'll get into some of those issues. All right, well, I don't want to keep you any longer. Thanks for that. And we'll see you then Tuesday for Pareto optimality.